Good morning, everyone, and welcome to the series on stem cell talks. And I'd like to welcome our speaker for today, Professor Monani from the United States of America. Welcome, and I hope you had a wonderful trip and you had a restive time yesterday. I would like to, at this point of time, introduce Professor Sangeeta Yoganathan. Sangeeta is professor in pediatric neurology, and she's an astute person in the area of neurometabolics. She has also done advanced training in Sick Kids Hospital in Toronto, in Canada, and has aspired to do great work in genetics. Over to you, Sangeeta, to chair the rest of the session. Thank you.
Good morning, everyone. Thank you, sir, for the kind introduction. It's my pleasure to introduce Dr. Umrao Monani, our invited faculty today. Dr. Monani is Professor of Neurology in Pathology and Cell Biology at the Columbia University Medical Center and the Director of Colleen Giblin Research Laboratory at the Medical Center. He is also a founder member of the Columbia University Center for Motor Neuron Biology and Disease. Dr. Monani received his PhD at Ohio State University School of Medicine, and he has spent many years engaged in the study of neuromuscular disorder, particularly the spinal muscular atrophy. He was amongst the first to develop viable SMA model mice, and this work has informed SMN repletion treatments for the human disease. Most recently, Dr. Monani has turned his attention to the Bldg deficiency syndrome, and he has shown that gene replacement strategies are highly effective in treating a mouse model of the human disease. These studies also revealed novel brain microvasculature defects resulting from Bldg deficiency syndrome. Currently, Dr. Monani's lab is engaged in exploring the feasibility of translating the preclinical gene replacement findings in his lab into a viable treatment for the human condition. Dr. Monani has received several awards, including the American Academy of Neurology Young Investigator Award, the Sanofi Innovator Award, and the 2023 Humanitarian Award from the Hope for Children Research Foundation, and the Darryl C. Vivo endowed professorship at the Columbia University. His lab is dedicated to the study of rare pediatric neurological diseases with a view to eventually treat them. So a warm welcome to Dr. Monani today, and the stage is his.
Thank you very much, Dr. Thomas, for hosting me over here. Thank you, Sangeeta, for the warm introduction. You know, I remember as a young scientist sitting in the audience and listening to a senior professor talk about his or her research, and I guess I'm fairly senior now, and I've done a lot of traveling and given presentations such as these in the United States, in Europe, and in India as well. And I do have to say that the hospitality that I am accorded when I come here to India is absolutely unparalleled. And so for that, there's a great warmth that I feel when I come and visit, even though I'm not acquainted with many of you people. The hospitality that you people have accorded me is something that brings me back time and again to places like CMC Vellore. So thank you very much for that. So I should, in the same breath, express my appreciation to Drs. Yoganathan and Matthews for hosting me here today. It's a great pleasure to be here in Vellore. I must admit, my first visit to the city, and to have been invited to one of the preeminent medical centers here in India. I do have to make an admission, and that is that I am not a clinician, but as Sangeeta said, the work that we conduct in my lab is carried out with the express purpose of developing novel treatments for human disease. And so of particular interest is infantile onset neurological conditions. And today, I'm going to focus on one of these that we've studied in the lab for a long time, the disease that you see there up on the screen, Bldg deficiency syndrome. Now, before I go on, let me just put up this slide. This sort of gives you a breakdown of the various parts of my presentation today. So I will start off with an overview of GLUT1 deficiency syndrome. I will then move on to testing a potential means of treating GLUT1 deficiency syndrome, because as I said, we study these diseases to understand the basic biology of these conditions, but really with the ultimate view of developing novel treatments for diseases with unmet medical needs, and this is one of them. Within this section, I will talk about a mouse model of this disease. This is an important preclinical tool that we have used in the lab for many years. I will talk about testing gene therapy for GLUT1 deficiency syndrome using this particular mouse model and the various outcomes that we look at in this particular mouse, including having used the mouse to identify what we think is a truly novel pathology in this particular disease, and one that I think begins to explain, at least in part, the brain dysfunction that is characteristic of this particular disease. I will then move on to defining when, during the course of the disease, it's important to initiate a treatment in order to make certain that that treatment is effective. And finally, then I will talk about defining when, or I should say where, in other words, what cells one really needs to focus on in order to ensure that the treatment is an effective one. And then I will end with a summary slide. I should just go back over here, and you might be wondering what this montage on the title slide is. And so this is a section from a mouse brain cortex, and the pink structures that you see over here are brain capillaries, and these blue spider-like structures are brain astrocytes, and the significance of these structures will become clear to you in just a bit. So moving on to the disease subject matter for today, and that is GLUT1 deficiency syndrome. So what is this disease? It's a rare disease, so I wouldn't be surprised if many of you folks haven't heard about this disease. And so as this particular slide elaborates, GLUT1 deficiency syndrome, also known as Vivo disease or glucose transporter 1 deficiency syndrome, it's a rare but life-altering neurodevelopmental condition that is caused by heterozygous mutations or a haploinsufficiency of this gene known as SLC2A1, which we also refer to as GLUT1, and therefore reduced levels of its translated product, the GLUT1 protein, which is what you see here on this Western blot. The classic GLUT1 deficiency syndrome disease phenotype becomes apparent in infancy or in early childhood, and it's characterized by epileptic seizures, cognitive impairment, developmental delay, and a complex movement disorder that combines elements of ataxia, dystonia, as well as spasticity. And I have a short video that I'll play for you that illustrates that. And then more recently, the GLUT1 deficiency syndrome disease phenotype has been expanded to include hemolytic anemias and exercise-induced dyskinesias. And when one takes that into account, as well as recent reports suggesting that GLUT1 mutations underlie a quite significant proportion of the idiopathic generalized epilepsies, then one is led to believe that this is not an ultra-rare disease, as was originally thought. We think that the prevalence in the United States is anywhere between 6,000 and 12,000. And since GLUT1 deficiency syndrome has no ethnic bias, one can use these numbers to extrapolate the prevalence of the disease around the world, and indeed in a country like India. But despite this quite complex disease phenotype, if one had to pick one signature feature of this disease, it would be that these patients have abnormally low levels of cerebrospinal fluid, or CSF glucose, a condition that is known as hypoglycorrhea. And it took me many years to, it's an easy word to say, but it took me many years to figure out how to spell it. And I should quickly point out that the low CSF glucose is in the context of normal levels of glucose in the blood. So hypoglycorhea, but in the context of normoglycemia. This protein is widely expressed, but it's especially abundant in the endothelial cells of the brain microvasculature. And it is here that GLUT1 facilitates the diffusion of glucose from the circulation into the brain parenchyma to satisfy the energy requirements of the vast neuronal circuitry of the brain. And I should say that glucose is by far the preferred energy source for the brain, and the depletion of that is the reason why you have this disease phenotype. The molecular genetics of this disease were described way back more than 35 years ago by Vivo and colleagues, who actually recruited me to Columbia University and then convinced me that this is a disease that I ought to work on. But sadly, there is still no optimal treatment for this condition. The current standard of care involves administering to these patients a high-fat, so-called ketogenic diet, which supplies the brain with an alternate fuel source, ketone bodies, but which is only partially effective at best. And this next slide over here really illustrates and highlights the caveats associated with the ketogenic diet, the shortcomings associated with this ketogenic diet. And this was compiled by following between 30 and 50 patients, all of whom were on the ketogenic diet. I mean, they were followed out for a decade of life, sometimes 2 decades of life. And as you can appreciate over here, despite the fact that these patients were on the ketogenic diet, almost a quarter continued to experience seizures. 60% continue to exhibit this paroxysmal dyskinesia, which is this movement disorder that is characteristic of this disease. And almost 3/4, 72%, continue to experience dysarthria. And so we really need an alternative to this ketogenic diet. Even patients who are on the ketogenic diet can be quite severely affected. And there are some patients who just are intolerant to the ketogenic diet. And that cannot be made much clearer than it is on 3 short videos that I have on this particular slide. And switch from this. Here you will see 3 videos of 2 patients. The first video is of a girl about 12 years old, and her mother came to the neurology, the pediatric neurology clinic at Columbia, and she was absolutely desperate because she has tried the ketogenic diet in various forms, modified Atkins diet, a 3-to-1 fat to carbohydrate ratio, a 4-to-1 fat to carbohydrate ratio, and just nothing has worked. And you'll see that in the video that I will play for you. In the second video, you will see the movement disorder in the same child, and in this particular video, the last one, you will see a child who also came to the pediatric neurology clinic. She's about 2.5, 3, and she is experiencing an absence seizure. So let me play this first video for you. See. So it's actually quite distressing to watch, I have to say. So she's undergoing a convulsive seizure. And as I said, again, despite the ketogenic diet, and this is one of the reasons why this family is so distraught. These kinds of seizures, you can imagine, really affect the quality of lives of these particular patients. Here you'll see the movement disorder in the same child. You can see that she has these knee pads on. Let me just oops. If she doesn't have the knee pads on, she falls and she'll hurt herself if her parents are not holding her hand. Notice the abnormal gait, and so this is another characteristic feature of glucose deficiency syndrome. It usually happens, it can happen very early on in life, but it's usually preceded by the seizure activity, and the movement disorder persists for the duration of the lives of these patients. And again, if you're constantly falling down, that really affects the quality of lives of these patients. And over here, you'll see this child experiencing an absence seizure. You'll see that she's sort of becomes completely oblivious to what is going on around her. So you can hear in the background her mother is trying to get her attention, but she's just staring into space. She's completely oblivious of what is happening. So you can see that this lasts for about 10 seconds or so, and this can happen dozens of times a day. And you can imagine that this also affects the quality of lives of these patients. And so, this disease is really one with an unmet medical need. And so we've spent a lot of time trying to address the needs of these patients to come up with an alternative to the ketogenic diet, one specifically that addresses the root cause of this disease. So what have we been doing? But before I go on to that, let me very briefly just go over this particular slide because this is a slide that describes the preclinical tool that we have been using in the lab to try to develop this treatment for glucose deficiency syndrome. So in 2006, the Vivo lab generated a mouse model of this condition, and I was fortunate to advise them on that. And while it might be argued that this mouse model does not exhibit as severe a phenotype as the classic glucose deficiency syndrome patient, it is nevertheless in many respects faithful to the human disease. So let me just put the pointer back on. So the folks on Zoom can see what I'm pointing at. So it expresses reduced levels of the glucan protein, and that's because it's haploinsufficient for the murine version of the SLC2A1 of the glucan gene. It exhibits a motor phenotype as assessed on a rotor rod, and more about that in just a moment. The brains of these animals are deficient in their ability to take up glucose, and the brain is also somewhat diminished in size, which is reminiscent of the acquired microcephaly that one sees in the human patient. And last but not least, these animals have greatly reduced levels of CSF glucose, which is akin to the hypoglycorrhea that one sees in the human patient. And so given the cause of a glucan deficiency, which is essentially reduced levels of glucan, and our goal of trying to develop an alternative to the ketogenic diet and at the same time address the root cause of this particular disease, we felt that glucan repletion is an intuitively appealing proposition. It worked very nicely in the case of another disease that we've studied in the lab that some people refer to, spinal muscular atrophy. And so here's the experimental strategy summarized in one half of one slide. It took advantage of AAVs, adeno-associated viruses, which some of you might be familiar with because of their success in the treatment of spinal muscular atrophy. And so what we did was to package the glucan gene into this AAV, and we administered it into our mouse model, and then we assessed for therapeutic outcome. But before we began that, we asked some simple questions. The foremost of which was, does glucan gene repletion actually raise levels of glucan in this case, we look at fibroblasts from a patient with glucan deficiency syndrome. And these were sort of proof of concept studies that we did before embarking on this more complicated set of experiments. And so this is a representative Western blot, and you can see that the fibroblasts from the patient with glucan deficiency syndrome express quite low levels of glucan compared to fibroblasts from a healthy control. We can also see that irrespective of whether one uses a murine copy of the glucan gene or a human copy of the glucan gene, when you transduce these fibroblasts with either copy of the glucan gene, the levels of glucan do indeed increase. And so then we asked, does this increase in the glucan protein in the fibroblasts, does that actually result in an enhanced ability of these fibroblasts to take up glucose? And that was important because you don't just want absolute levels of the protein to go up. You want to make certain that that protein is functional. And so this was a functional assay that we carried out. And you can see that the fibroblasts that have been transduced with these constructs now have an enhanced ability to take up glucose. And finally, and this was assuming that one would have to focus on the brain microvasculature where these endothelial cells reside and where glucan is expressed at high levels. We asked ourselves whether this AAV that we were using transfused the brain microvasculature efficiently. And in this case, one is referring to AAV9. It's a particular serotype of adeno-associated viruses. And we selected AAV9 out of force of habit, really, for want of a better way of explaining it. AAV9 had been used successfully in the context of spinal muscular atrophy all the way into the clinic. And in fact, it's used in the clinic right now. And so we figured AAV9, and this is obviously the mouse brain microvasculature. And so what you're looking at over here are sections through the cortex of a mouse brain. And what we did was we administered an AAV EGFP, green fluorescent protein construct into the circulation of these animals. These squiggles that you see are brain capillaries. The signal in red represents endogenous GLUT1 protein, and we co-stained with an antibody against GFP because these animals were administered an AAV GFP construct. And you can see that there's fairly good overlap between the green and the red, and that suggested to us that AAV9 is a reasonable vector or serotype to use. And so we said, let's move forward. We packaged the GLUT1 gene, in this case, the murine GLUT1 gene, into AAV, and we administered it to our mouse model of GLUT1 deficiency syndrome. And then we asked ourselves, okay, does this mitigate the disease phenotype? And the first thing that we looked at was the movement defects that are also seen in this mouse model. And we focused on two. One was the rotor rod test, which is carried out on a contraption similar to this one over here. And essentially what you do is, so this rod over here rotates at different speeds. And we put the mice on that and record the latency with which they're able to stay on. And that is a nice assay that has worked very well for us the time that we've studied GLUT1 deficiency syndrome. And these are the results of the assay that we carried out. The bars in red represent mutants that were administered just vehicle. The bars in black represent the performance of wild-type healthy controls, and the bars in gray represent the performance of mutants that were administered this AAV GLUT1. And you can appreciate that the mutants that were administered just the vehicle alone perform very poorly compared to their wild-type healthy control littermates. In striking contrast, you can see that the mutants that were administered the AAV now perform significantly better. And we found this to be the case at all of the different time points that we examined. And then in the second assay, the vertical pole test, where we place the animal on a vertical pole like so, and then record the latency with which they're able to reorient themselves, turn around, descend it without falling off. The results were very similar to the results that we found in the rotor rod test. In other words, the mutants that had been administered the AAV now performed akin to their wild-type healthy littermate controls, and significantly better than mutants that had been administered just the vehicle. And so this told us, this was very promising. This told us, yes, indeed, at least one aspect of this particular disease, these motor defects, are mitigated by AAV-mediated GLUT1 repletion. So we next asked, okay, so this is great, but still early days. Does this correspond to an actual increase in GLUT1 protein? Because that was the mechanism of action that was predicted. And so we assayed for levels of GLUT1 in the brains of our 3 cohorts of animals, which is what you see in these 3 top panels. This first graph represents the quantification of the GLUT1 transcript in our 3 cohorts of animals. And over here, you see it represented the Western blot. This is the quantification of the Western blot. So in red, or I should say red represents mutants that were administered just vehicle. Blue represents the wild-type controls, and green represents the mutants that were administered the AAV. And you can see a big difference in GLUT1 transcript levels in the brains of the animals when they are mutant and have been treated just with vehicle alone compared to wild-type animals. And you can see a significant increase in the levels of the transcript when you treat the mutants with AAV GLUT. And we see corresponding increases in GLUT1 protein, and here's the quantification. So as I alluded to earlier, GLUT1 deficiency syndrome patients, as well as these model mice, suffer from hypoglycorrhea, reduced CSF glucose, micrencephaly, which is equivalent to this acquired microcephaly that one sees in the human patient, and then an inability, or I should say, an inefficiency with which the brain is able to take up glucose for the needs of the functioning of the cerebral neurons. And so we found that all of these had also been mitigated by the GLUT1 gene repletion. So let's focus on the CSF glucose. Again, the red represents mutants treated with vehicle alone, blue represents wild type, and green represents those mutants treated with AAV GLUT. So you can see a big reduction in CSF glucose in mutants that have been treated with vehicle alone. And when you treat them with AAV GLUT, the hypoglycorrhea is significantly mitigated. No change in levels of blood glucose, as I said, hypoglycemia, but in the context of normal glycemia. But because CSF glucose levels increase, CSF to blood glucose ratios also increase, which is quantified over here. And consistent with this, we found that microencephaly, the microencephalic phenotype, had in this case essentially been prevented, which is what you see over here. Body weight, at least in the mouse model, doesn't change. In certain patients, you do see a decrease in body weight, but in the mouse model, it doesn't. And so because brain size is normalized, brain to body weight ratios are also increased in these mutants that are administered AAV GLUT, not shown over here, but reported in a publication that we put out many years ago in Nature Communications. This is the result of a PET imaging study following administration of a radio-labeled glucose analog into the circulation of our 3 cohorts of animals. And what I'd like to draw your attention to is the fact that the intensity of the signal in the brain of the mutant that has been administered AAV GLUT1 is now almost equivalent to the intensity of the signal in the brain of the healthy control, and much greater than the intensity of the signal in the mutant that was administered just vehicle on. Here's the quantification, suggesting that this has also improved the ability of the mutant brain to take up glucose. In parallel, we found that the seizure activity that we see quite characteristically both in the human disease as well as in the mouse model had been mitigated, and that's what you see over here. So these are results from some EEG studies that we carried out, and you can see that in untreated animals or in vehicle-treated animals, you see lots and lots of seizures in the form of these abnormal spike wave discharges. These are EEG traces from a healthy control, and you can see that treating with AAV GLUT now results in EEG traces that are much more like the EEG traces in the wild type animal. And here's the quantification. So this was certainly very comforting to see, but perhaps not overly surprising given the root cause of GLUT1 deficiency and essentially what we were doing over here, which was to restore levels of the GLUT1 protein. What was much more interesting, and at that point in time, and continues to inform the cellular and anatomical basis of this disease, was discovering that GLUT1 deficiency results in what we term as a diminutive brain microvasculature, and that's what you're seeing over here. So these are thalamic sections from our 3 cohorts of animals that have been stained with lectin in order to visualize the brain capillary network. And what you will appreciate immediately is that compared to the complement of brain capillaries in the wild type animal, you have far fewer capillaries in the GLUT1 deficiency syndrome mutant, and they look rather fragmented. In contrast, when you administer the AAV GLUT to the mutant, you can see a restoration of the brain capillary network, and here is the quantification. Now, this was quite a surprise to us, and I remember when we sent our paper out for review, I think the reviewers were a bit skeptical, and they said, well, you've looked at this in animals that have been euthanized. This is all perfused tissue. Can you show this in the live animal? And so we did some live imaging, and consistent with what we found on the last slide, again, we found that the complement of brain capillaries in the mutant were significantly reduced. And so what this suggests is that there is an arrest in brain angiogenesis. You can see fewer capillaries over here compared to what you see in the wild type animal. And when you treat with AAV GLUT, you are able to restore the complement of brain capillaries, and here's the quantification. Even more striking are 3 short videos of this particular experiment where you can actually see the brain capillaries emerging as we image them using 2-photon microscopy. And that's over here on my next slide. So let me play these videos for you. And so you will see fewer capillaries over here, and the intensity of the signal in the capillaries is also reduced over here compared to what you see in the wild type animal. And there's a restoration of these capillaries and mutants that have been administered AAV-GLUT1. And I think what is very comforting to us is finding that what we discovered in this mouse model now appears to be true of the human patient as well. These models are only as good as how they reflect on what is going on in the human patient. GLUT1 deficiency syndrome patients have a normal lifespan. So coming by brain tissue from a GLUT1 deficiency syndrome patient is difficult, but we have managed to look at a couple of brains, and we have been comforted to find that this particular phenotype that we have observed in the mouse model does appear to be reflected in what is happening in the human patient as well. And I could go into the reasons why we think that we have this diminutive brain microvasculature towards the end of my talk. And so, by at least a half dozen outcome measures, we have shown that GLUT1 repletion prevents and or reverses the GLUT1 deficiency syndrome disease phenotype in this mouse model of the condition. And that obviously raises a great deal of optimism for the treatment of the human patient. I mean, we went through this exact same sequence of events when we were developing a treatment for spinal muscular atrophy. And as you're probably aware, we now have these treatments for spinal muscular atrophy. I have no doubt that this particular strategy would also work in the context of GLUT1 deficiency syndrome. I mean, obviously there would be challenges along the way, and we can discuss them if you're interested. But I think the proof of concept studies are quite compelling. The strategy, in a sense, is kind of what we term a no-brainer. You reduce GLUT1, you restore GLUT1, and you should prevent the onset of the disease phenotype. So along these preclinical experiments that I just presented to you, we were also very interested in addressing another very important question from the point of view of developing a therapy, and that was the requirements of this GLUT1 protein. So when is the protein required? When do you have to restore the protein in order to prevent the onset of the disease phenotype? But before I go on to that, let me very briefly touch upon the consequences of GLUT1 haploinsufficiency and this. Let me just put the pointer back on. Yes, and this diminutive brain microvasculature on the state of the blood-brain barrier, the integrity of the blood-brain barrier. And we were interested in addressing this question because of some reports that we had found in the literature to suggest that if you drastically reduce GLUT1 down to 10% of wild-type levels, then at least in a fish model of this disease, you compromise the integrity of the blood-brain barrier. It becomes leaky. And if that is really happening in the human disease, you want to know that. And so we looked at our mouse model, and I should quickly point out that we made certain that the mouse model, to the best of our ability, reflects the human condition. And the most important aspect of that is to ensure haploinsufficiency of GLUT1. Okay, so you reduce GLUT1 down to 50% of wild-type GLUT1 levels. And when one does that, what we found was that what had been discovered or observed in the fish was not what we found in our mouse model. And so this is the result of just one of these experiments. Essentially, what we did was we introduced into the circulation of these animals a labeled IgG, and then we looked for extravasation, meaning to say leakiness of the labeled IgG into the brain parenchyma of the mutants. And you can see that the pattern in the mutant is more or less identical to what you find in the wild-type animal. If, on the other hand, you take either one of these animals, in this case, we took the wild-type animal, and then you subject the wild-type animal to a known disruptor of the blood-brain barrier, such as kynic acid, you can see copious quantities of the IgG now leaking out into the brain parenchyma and showing up as these green puncta. Again, we wanted to make certain that this was not something that was specific to labeled IgG, and so we substituted labeled IgG with labeled BSA, bovine serum albumin, which is also a large molecule, we then substituted the IgG with labeled biocytin, which is a much smaller molecule and would therefore increase the sensitivity of the assay. Sometimes these large molecules, even though the blood-brain barrier is leaky, don't come out because of the large nature of these molecules. Biocytin is much smaller. And in every case, the result was identical to what we found over here. What is more, in the human patient, we have not found any evidence of an increase in serum proteins in the CSF of these patients. And so as far as we can tell, despite what has been observed in the fish model, where you have a reduction, a dramatic reduction, 90% reduction, in the human disease, in the context of GLUT1 haploinsufficiency, the blood-brain barrier seems to be fairly intact. So going back to this question of the requirements for the GLUT1 protein, you see, GLUT1 deficiency for the most part is diagnosed only once the patient becomes symptomatic. Kind of akin to spinal muscular atrophy before the advent of newborn screening. My guess is that in developing countries, spinal muscular atrophy continues to be diagnosed again when the patient becomes symptomatic. And so we were interested in asking, the pre-symptomatic treatment worked very nicely in the mouse model. What happens if one treats during early symptomatic phases of the disease or late symptomatic phases of the disease? Because you can easily imagine that these patients come in when they are symptomatic. And so we want to know if one begins GLUT1 gene depletion, would we get the same benefit as we did when one begins a treatment pre-symptomatically? And so in the context of this particular mouse model, early symptoms become apparent at about 2 weeks of age, and full-blown symptoms are observed between 6 and 8 weeks of age. And so what we did was we treated an additional 2 cohorts of animals, one at 2 weeks of age, representing this early symptomatic phase of the disease, and another cohort at 8 weeks of age, representing this late symptomatic phase of the disease. And then we examined them for the exact same outcome measures that we had examined in our cohort of animals that had been treated as neonates. And here are the results. Now, this is a rather busy slide, but the bottom line is this. If you treat when the animal is fully symptomatic, the benefit is dramatically reduced. And this is despite robust AAV-mediated transduction, despite an significant increase in GLUT1, and a significant increase in CSF glucose. And so in contrast to that, when you begin treating during an early symptomatic phase of the disease, you do see benefit, but it's not quite as dramatic as the benefit that you see when you begin treating pre-symptomatically. And so let's just very briefly go over this. In the animals that were treated at 8 weeks of age, it's the brown bars and the brown symbols that are the ones to focus on. So you can see an increase in GLUT1 transcript in the brains of these animals, corresponding increase in GLUT1 protein, increase in CSF to blood glucose ratios, no benefit in the rotor-rod test, meaning to say that these motor defects persist, and the microencephalic phenotype remains quite obvious. In contrast, see what happens when you treat at the early symptomatic phase of the disease. It's the green bars and the green symbols that are ones to focus on again. An increase in GLUT1 transcript, increase in GLUT1 protein. You see an increase in CSF blood glucose ratios, but here we do see an improvement in motor performance, and the microencephalic phenotype has essentially been prevented from occurring. And so consistent with this, when we examine the brain microvasculature of these cohorts of animals, here's what we found. So a couple of controls, the brain capillary network in wild-type animals. In mutants, you can see a big difference. When you treat at 8 weeks of age, there's no difference between the pattern that you see over here and the pattern that you see over here. When you treat during pre-symptomatic phases of the disease, the brain angiogenesis is more or less normalized, and you see an intermediate phenotype when you begin treatment during early symptomatic phases of the disease. And here's the quantification. So what this suggests is that there is a critical early requirement for the GLUT1 protein, And in a sense, this is perhaps not that surprising given the infantile onset nature of this particular disease. This begins to inform how we would treat these patients when they come into the clinic. And obviously, the caveat is that what we are seeing in the mouse model is reflective of the human disease. I'm optimistic that what we are seeing over here is truly reflective of what is happening in human disease. We saw this in the case of spinal muscular atrophy as well. And so the take-home message from this appears to be that there is this early requirement for the glucan protein. Now, in more recent work, we've corroborated this finding using a somewhat different GLUT1 protein depletion strategy. And that's described over here. And in a sense, we were also at the same time trying to answer this very important question. What are the effects of losing a therapeutic vector over time? So assuming that we adopt GLUT1 gene therapy in the clinic, one would imagine that transfused cells eventually turn over. So what happens when these transfused cells turn over and you essentially return the subject to a GLUT1 deficiency syndrome state? And so we did this experiment in order to at least partly address this question. So this was a genetic experiment, as you can see over here. What we did was we took animals that were floxed for the SLC2A1 of the GLUT1 gene. And by floxed, I mean that they contain, or they harbor, it's represented by this FLFL, they harbor an allele or two alleles of the murine SLC2A1 of the GLUT1 gene that can be inducibly inactivated. And we bred them to animals that expressed a tamoxifen-responsive, ubiquitously expressed Cre-ER transgene. These floxed alleles can be inactivated using Cre. What we did was we selected for animals that were transgenic for Cre-ER and were heterozygous for the floxed allele. And then what we did was we induced haploinsufficiency in these animals at 3 different points in time during the course of their lives. At postnatal day 2, which represents a neonatal phase of life, at 2 weeks of age, which represents a juvenile phase of life, and then at 8 weeks of age, which represents an adult stage of life. And then we asked, and this was with tamoxifen, and then we asked, how does this affect the health of these animals? So first off, we were able to show that when you deplete GLUT1, or I should say, make GLUT1 haploinsufficient at postnatal day 2, it is essentially equivalent to a constitutive GLUT1 haploinsufficient state, meaning to say that it's equivalent to inducing haploinsufficiency from the point of time of conception. And the evidence for that is on this slide and on my next 2 slides. So first off, we were able to show that we had robustly reduced levels of GLUT1 transcript. This is reflected in reduction of GLUT1 protein, which is quantified over here. This resulted in obvious hypoglycorrhea, microencephaly. When we looked at the brain, I should say before that, the rotor rod performance movement disorders, we found that these animals performed very poorly on the rotor rod test, and the frequency with which we saw seizures in these animals had greatly increased. And consistent with this, we found that brain angiogenesis had been arrested, which is what you see over here. Here is the quantification. And so what happens when one depletes the protein when the animal is at a juvenile stage of life and when it has turned into an adult? The results of that are shown on this slide and the next couple of slides. So once again, we were able to show, yep, we've managed to reduce GLUT1 down quite significantly, transcript levels, protein levels. Here's the quantification of the protein. Again, we were able to show reduced levels of CSF glucose. CSF to blood glucose ratios also change. But look what happens when one assays for motor performance and makes brain size measurements. On the rotor rod, we found that depleting the protein during early symptomatic phases of the disease did actually result in significant impairment. And that's represented by the bars in, I guess, beige. On the other hand, animals that were depleted of the GLUT1 protein, once they had attained adulthood, they performed fairly normally on this rotorod. And consistent with this, we found that the microencephalic phenotype had been triggered in the animals where the protein was depleted during juvenile stages of life, whereas it wasn't obvious at all in animals that had been depleted of the protein at 8 weeks of age. And consistent with those findings, you can see over here that the brain microvasculature in animals that have been depleted of the protein at 8 weeks of age doesn't change, whereas we do see a somewhat reduced brain microvasculature in animals that have been depleted of the protein at 2 weeks of age. And what this suggests is that once the animal has attained adulthood, meaning to say, once the animal has gone through a point in time when the GLUT1 protein is truly critical for brain function, for establishing brain capillary networks, for establishing brain neuronal networks, then depleting GLUT1 protein appears to have fewer adverse effects. And in a sense, again, this is sort of intuitive and reflective of what we found in the case of spinal muscular atrophies as well. But where might this GLUT1 protein, where in the brain might this GLUT1 protein be really important during this early neonatal phase of the lives of these mice, and most likely in the case of patients as well? And so to answer that question, first of all, we were aware that GLUT1 is expressed at high levels in the brain microvasculature, specifically in the endothelial cells of the brain microvasculature. But GLUT1 is also expressed at high levels in brain astrocytes. And at that point in time, and this goes back to studies that we published in 2021, so these were studies that had been initiated much earlier, we hypothesized that the endothelial cells of the brain microvasculature are a critical cellular site of action of the GLUT1 protein. The experimental strategy that we used to test this hypothesis was fairly straightforward and is essentially summarized in this slide over here. So once again, this is a genetic experiment. Again, we used the animals that were floxed for the SLC2A1 allele. This time we crossed them with this Tie2-Cre transgenic animal. Tie2-Cre being a Cre that is expressed in the endothelial cells of the brain microvasculature. And what we were trying to do over here was answer this important question: does selectively depleting GLUT1 in the brain endothelia, is that sufficient to trigger GLUT1 deficiency syndrome? And so we obtained these animals that were transgenic for the Tie2-Cre and floxed for the SLC2A1 allele. And then we asked ourselves, is this sufficient to trigger the disease? And it is. So before we started assaying for the various outcome measures, we asked ourselves the simple question: has this actually resulted in selective depletion of GLUT1 in the brain capillaries? And you can see over here that it does. This is a representative Western blot showing that GLUT1 has been depleted in the brain capillary fraction, whereas in the neuronal fraction, in the neuropil fraction, which consists of brain neurons and astrocytes, you can see that there's essentially no change. So is this sufficient to trigger GLUT1 deficiency syndrome? It is. You can see that rotorod performance is affected. Hypoglycorrhea is triggered. One sees evidence of the microencephaly, and one sees a big increase in seizure activity. Here are two spike wave discharges in an EEG conducted on the mutant. This is a normal EEG trace. And again, consistent with this, what we found was that the brain microvasculature, or brain angiogenesis, I should say, was arrested. And this also told us that this brain angiogenesis, this arrest of brain angiogenesis under conditions of GLUT1 deficiency syndrome, are really a cell-autonomous effect. And here's the quantification. So what is the basis of this diminutive brain microvasculature and this arrest in brain angiogenesis that I have been going on about for the last 40 minutes or so? Well, as it turns out, brain angiogenesis is critically dependent upon a particular type of endothelial cell lining the brain microvasculature, an endothelial cell known as an endothelial tip cell. And these endothelial tip cells are found at the leading edge of brain capillaries. What they do is they sense the brain microenvironment at a point in time when the brain capillary network is being established. In the mouse, it happens during the first 2 to 3 weeks of age. My guess is in humans, it probably happens in the first like 5 to 6 years of life. Okay, so they sense the brain microenvironment, and they put out these finger-like processes that we refer to as lamellipodia or filopodia, and those then pioneer new blood vessels. And what we found, interestingly, was that there were far fewer of these endothelial tip cells in our glutamate deficiency syndrome mutants, specifically in these mutants. And that's what you're seeing over here. So by now, you should be quite familiar with these kinds of photomicrographs. This is the capillaries from a control animal, capillaries from a mutant. You can see far fewer capillaries over here. But what you also see, and what I've tried to illustrate over here, these images were taken from animals where brain angiogenesis is actively taking place. You can see these endothelial tip cells. In the control, you can see 1, 2, 3, 4, 5 of them, okay? In the mutant, you do see them, but you can see that they are stunted. And this is better appreciated in high magnification images, which you see over here. So this is a wild-type endothelial tip cell. Note the profusion of these lamellipodia or filopodia, and they extend quite far out from the cell body. This is the mutant equivalent. It does exist, but you can immediately appreciate the fact that it is quite small, and these filopodia or lamellipodia are fewer in number and quite stunted. And here's the quantification, the green versus the orange. So we were interested at this point in time in asking, is this a line-specific effect? I mean, is it specific to animals where we've depleted glutamate protein in the brain endothelial cells? And so what we did was we looked at animals that were constitutively haploinsufficient for the glutamate protein, which is essentially the condition that you find in the human patient. That's represented by the bars in red. And you can see that there's no difference between the constitutive haploinsufficient animals and those that have been depleted of glutamate, specifically in the brain endothelial cells. And what's more, if you take these animals and you subject them to AAV-mediated glutamate gene repletion, you can prevent the endothelial tip cell pathology. Here's some additional quantification of the defects that one finds in these endothelial tip cells. So, in summary, we've shown that glutamate repletion is a promising means to treat glutamate deficiency syndrome in a mouse model of the disease, and that such repletion may be affected by gene replacement. As I said earlier, this obviously raises a great deal of optimism for the treatment of the human patient. Along these lines, we've shown that restoring glutamate pre-symptomatically provides the most significant protection against glutamate deficiency syndrome. When you begin treatment when the subject is fully symptomatic, the benefit is much more muted. And when you begin treating at an early symptomatic phase of the disease, you have an intermediate phenotype. This is again quite similar to what happens in spinal muscular atrophy and consistent with the fact that this is an infantile onset neurological disease. On an anatomical and perhaps a more basic level, we've shown from our studies that glutamate deficiency syndrome results in this diminutive brain microvasculature. And you can easily imagine why this results in this kind of a disease phenotype. Remember, this is a disease of haploinsufficiency. There are many diseases where you lose one healthy allele, and as a carrier, you're completely fine. But this diminutive brain microvasculature, what it does is, first of all, it reduces the number of capillaries in the brain. And so the amount of nutrients that can go into the brain are reduced. And secondly, those brain capillaries that do exist have reduced amounts of glutamate protein. So in a sense, and as we say in the US, the brain undergoes what is known as a double whammy. And so it's not surprising that this haploinsufficiency then results in this disease phenotype. And what we've shown is that this is an endothelial cell autonomous effect that most likely originates in these endothelial tip cells. So I would be completely remiss if I didn't put up an acknowledgement slide. These are the folks in the lab who carried out the work that I presented to you these last 40 minutes. Here are our funding sources, without which none of this would have been possible. I thank you very much for your attention, and I'd be most pleased to take questions. Thank you.
Great study, very nice study, impressive. So my question is, how much of protein required? If you look at your AAV9 transduced cells, they seem to have much better protein than your wild type. But when we look at the functional effects, I mean, this AAV subjects, they are not catching up to the wild type, right? So are you delivering too much of protein?
I think it's unlikely. There might have been a couple of Westerns where you might have seen protein levels that somehow were higher than levels in the wild type animal. But in the vast majority of instances, actually, I would argue that you're probably not quite getting to wild type GLUT1 levels. And I think that explains why you don't completely normalize the animal. By many measures, I think, for instance, if you look at the microencephalic phenotype, that has been completely arrested. The diminutive brain microvasculature, when you start treating early, that has been prevented. I think a lot of this also has to do with the challenges with AAV-mediated treatments. I think it would be naive to expect that you get to completely wild-type levels. You might get to supraphysiological levels in certain cells and certain tissues. But what you really want to do is you want to, if you argue that it's the brain endothelial cells that are truly important, you want to make certain that all of the brain endothelial cells are equivalently transduced and begin to express at least wild type or close to wild type levels. But that rarely happens. You probably have some proportion of these cells that have been truly restored to wild type levels, whereas others are not. But I think if you transduce sufficient numbers of these cells, you get a very good outcome.
What happens to the homozygous now?
It's embryonic lethal.
Lethal. Yeah, right. So this may not be a rare disease, right? In case we are losing the homozygous mutant, it may not be.
I mean, GLUT1 is absolutely fundamental, and you can imagine why. I mean, if glucose is the main energy source for the brain, you completely deprive the organism of GLUT1. At least in the mouse, the animals don't get to beyond about E9. And so the process of resorption in the mouse embryo probably begins at E6 or something like that.
Thank you.
Thank you so much for the incredible work and excellent talk. I have a question, like if you translate in the humans, like the age at onset of symptoms is highly variable, and there is phenotypic variability in humans. Like some of the variants can cause like a congenital onset of symptoms, like having congenital microcephaly with hemolytic anemias, because GLUT1 is expressed in RBC as well. Ranging to the adult, just manifesting with a mild ID and psychiatric manifestations. So if you want to start the treatment as a pre-symptomatic stage, like would you consider planning an genotypic characterization and like delivering the gene product, gene therapy in utero?
I don't think there's any evidence as yet for a requirement for wild type levels of GLUT1 in utero, unlike in SMA, where there seems to be accumulating evidence that there is an important requirement for SMN, even in utero. In glucose deficiency, as far as we can tell, the animals, at least, I mean, I'm talking about the mouse model, and my guess is it's probably similar to the human as well. Embryonic development is more or less normal. And then depending on the particular mutation that you have, the glucose mutation that you have, and the complement of genetic modifiers of GLUT1 or interactors of GLUT1 that you have in the background, you would probably need to either start treating very early after birth, or probably within the first year or so after birth. We're having this sort of debate in SMA, as you well know, and what we do know from the mouse model is that some of the brain pathology that we have discovered becomes obvious very early on. I would say that by about 2 weeks of age, one begins to see neuroinflammation in the brains of these animal models. Now, whether that is adaptive or maladaptive is currently unclear. My guess is it's probably adaptive in the first instance and then becomes maladaptive. And so I think the bottom line is that even though onset can be fairly variable, I think that even in the mildest individuals, if you look carefully enough, you will probably start seeing evidence of GLUT1 deficiency symptom disease characteristics fairly early on, even in the most mild patients. As the question is, can you catch them? I mean, these kinds of absence seizures. I mean, before I started studying GLUT1 deficiency syndrome, I mean, you see people staring into space. Is that a seizure? I mean, you think that he's just not paying attention, or she is not paying attention, and then when you do the EEGs, you find, oh, this is associated with abnormal brain electrical activity. And my guess is that you're probably going to see this. CSF glucose also is probably going to be reduced in those individuals. I think what's very interesting is, why is it that those patients develop or present with such mild symptoms? And I go back to something that I found fascinating and that convinces me. I think that this applies to all of the various diseases that we study. These genes don't work in isolation. These genes are part of pathways, okay? And so you have other genes that exacerbate the disease, and you have yet another family of genes that suppress the disease phenotype. And it's very, very important. I mean, this is like basic biology, but you can easily see that it has therapeutic implications down the road. But you've got to start somewhere. You identify these genes and you find, first of all, this belongs in the same pathway. And maybe this is a better therapeutic target than actually going for what is deficient in the disease. One such modifier, one could argue, is a long non-coding RNA that we have identified, okay? And you can modulate the expression of the long non-coding RNA, and that has an effect on GLUT1. In the case of SMA, we found a synaptic chaperone, because you would think that it has nothing at all to do with SMN. But our studies seem to suggest that it has everything to do with SMN. And so I think the quite mild phenotypes that you see in some patients, especially in families where you have the mutant gene being inherited in the child from a parent who you'd never know was abnormal until they come in for the clinician to take a look at the child. And then you find that, the child and the parent have the same mutation, but the parent got through life to that point in time fairly normally, whereas the child is quite severely affected. And it's the genetic background. It's been difficult to find a strict phenotype-genotype correlation in GLUT1 deficiency. That being said, I would say that in general, if you have microdeletions, you will generally have a more severe phenotype. If you have missense mutations, it's generally mild. If you have nonsense mutations, they lie somewhere in the middle of that spectrum. But I think that if you want to catch this early, we're going to have to expand newborn screening. And I think it's going to happen. I mean, when I started working on all of this, sequencing the entire human genome, that was a project in itself. Whereas now the cost of this has come down quite substantially. I think what makes it a bit difficult with diseases like GLUT1 deficiency is that there's no one common mutation like in spinal muscular atrophy. You just look for presence of exon 7. But that being said, also, I think that the sequencing and the power of the computational technology necessary to identify these mutations, it's increased by leaps and bounds now. What's quite interesting now is we have this cohort of patients, and they have a classic GLUT1 deficiency syndrome disease phenotype. No mutation in GLUT1. No, not in the coding region. And so what causes this? And we've had some of these patients sitting around for a long time. And one of the postdocs who recently came into the lab, he was doing the sort of thing when he was at Ames, and he said, "Can I take a look at them?" I said, "Okay, take a look at them." I mean, people have been looking at these at the sequences for a half dozen years. Nobody's been able to find anything. He sat down and he took a look at them. What he found was quite fascinating, which again begins to inform on the basic biology and regulation of these genes. One particular example that is particularly relevant is a child who has a classic GLUT1 deficiency syndrome disease phenotype, no mutation in the coding region, no mutation, nothing, 50 kilobases downstream, 50 kilobases upstream. When you look 100 kb upstream, you find a balanced translocation. What does that tell you? When you look bioinformatically, there is an enhancer that is lying up over there. That's really important because what it tells you is that there are elements way removed from a particular gene that have a very important bearing on the expression of that particular gene and therefore the function of that gene. Those are going to be more challenging to identify when you do these large-scale sequencing projects. But I think AI is going to help a lot with that.
Those cohort of patients, did they have biochemical evidence of hypoglycemia?
Absolutely, absolutely. What's also very interesting is, the Vivo lab had pioneered the red blood cell uptake assay. So while he was the director of the Giblin lab, that used to be routinely done. But what was found was the red blood cell uptake assay in that particular individual was fine. Yet the patient had hypoglycemia. Okay. So what it tells you is that GLUT1 is functioning differently in the red blood cells from the way it's functioning. Yes. Okay. And I mean, those are rare cases, but again, it in incremental fashion, it begins to inform the mechanisms underlying these diseases. And then you get to a certain point where you have a critical mass of data, and you say, now we can develop a novel treatment that completely changes the quality of lives of these patients. It becomes really effective. But you need, otherwise what tends to happen is, you make some superficial findings, you develop a treatment, and subjecting the patient to the treatment is better than not subjecting the patient to the treatment. But you have other side effects that occur and so on and so forth. You've got to refine the treatment, et cetera, et cetera. And that happens to a large extent. While one would like to make breakthroughs in the first instance, that often doesn't happen. It's an incremental process.
those mouse models who are haploinsufficient for SLC2A1, you assess the motor performance using the rotarod and the vertical rod test, and also like you looked at the microencephalic improvement by the stadium. Did you also look at the cognition and behavioral effects on these mouse models?
We are beginning to do that. What I've discovered through my research career is that the neuromuscular system, because this is based on our work on spinal muscular atrophy, is much better preserved or conserved, I should say, between humans and mice. Brain function is, I think, less conserved. And so you've got to develop fairly sensitive assays in order to determine whether there are these memory deficits, cognition deficits, etc., in the model mouse. But we are beginning to find evidence of that. The cohorts, the mouse cohorts have to be large in order to appreciate small effects. I mean, clearly in the human, there are these defects, and so, that's an ongoing project.
Since we already have amazing talk and work, absolutely. Since we already have, some kind of a treatment in the form of a ketogenic diet for these patients, and we do see that, 60% of them actually respond. Have you looked at, the mouse, models of, ketogenic diet and how it works in these mouse models? Have you looked at that?
That's a treatment. So that has been a goal for a long time in the lab. The mice are kind of on a ketogenic diet while they're nursing because of the high fat. We haven't empirically tested whether continuing that diet in the mice does benefit the animal or not. And it's not because we don't want to, it's just we don't have the bandwidth. You need to have a person dedicated to that particular project, and these are long drawn out projects. You've got to follow the mice out for a long period of time. What I can tell you is that my prediction is that certain pathologies, I mean, clearly the ketogenic diet, even under the best circumstances, does not prevent the onset of the disease. It's fairly good at suppressing seizure activity. It's pretty bad, I would say, at preventing the movement disorders. Those movement disorders, and I think that some of the brain pathologies are going to be less responsive to the ketogenic diet. And I think that's because ketone bodies feed into the TCA cycle, okay? And they replenish the constituents of the TCA cycle, but they don't do anything at all to replenish the glycolytic intermediates, specifically lactate. And lactate, it's becoming clear, functions well beyond just being a nutrient for the brain. Yes. It's involved in neuronal plasticity. It's
the worst of microencephaly, it can have a control on seizures alone, and again, like not in 100% cases.
I think one thing also should be mentioned and emphasized, and that is that, I mean, I'm biased. I sort of get hung up on one particular rare disease, and there are others who do that. I mean, you choose your particular rare disease that you're fascinated with for any number of reasons. But I do believe that GLUT1 is such a fundamental molecule that it impacts diseases well beyond GLUT1 deficiency syndrome. We are now beginning to explore the link between GLUT1 and Alzheimer's disease, because it's been shown for many, many years that the brain capillary network in AD patients is reduced. You have this sort of diminutive brain microvasculature. You have the brains are hypoglycemic, I should say, neuroglycopenic. And so this work that we've done on GLUT1 deficiency syndrome is now leading us towards exploring the relevance of GLUT1 in these other diseases. It's involved in retinitis pigmentosa, obviously diabetes, cancers. In cancers, what you find is that GLUT1 is upregulated, so you want to decrease the levels of GLUT1 in cancers. And so, our studies on this and the information that we glean about the biology of GLUT1 then becomes relevant to lots and lots of health conditions.
Any questions from the floor and any questions from anybody online?
This is excellent work. So you have mentioned in some cases there is no mutation in the gene coding regions or promoter, but in the distant enhances. So when you try to overexpress this gene using AAV, you need those regulatory elements to to activate that maximum.
Listen, we are ecstatic that we can prevent the onset of the disease using AAV GLUT1 treatment. But you're absolutely right, okay? There are all these elements that intricately control the particular gene. In this case, it's GLUT1, okay? I realized this some time ago. It is naive to think that you're essentially reverting or restoring what you'd find in the healthy state. Yeah, if you have these distant elements that intricately regulate a particular gene that you're interested in, and it's not there, what are the implications of that in the long term for these patients who have undergone this kind of treatment? Okay, and so I think we have to be cognizant of that and be prepared for these kinds of things to happen. Have it in the back of your mind. I mean, you don't let it paralyze you and say, you know what, okay, so we aren't able to put in this entire region into the patient. And so let's just not even bother with developing a treatment for this disease. But you've got to be cognizant of that. I mean, I appreciate what you're saying. You're absolutely right.
So one more question. So you put a spectrum of phenotype, mild to moderate to severe. So those patients has some alternative pathway to take the glucose inside the endothelial cells, or it's because of abundance of glucose in the cells determines the phenotype.
So glucose entries into the brain is essentially facilitated diffusion. And so it's based on a gradient. And so one of the things that I didn't mention is, at this particular juncture, GLUT1 repletion is not a reality. It's not a clinical reality. I mean, there is the aspiration that it will become one, but at this particular juncture, we just don't have it. So what do we do in the case of these patients who find the ketogenic diet intolerable or who are not benefiting from the ketogenic diet? So there is a substance known as diazoxide that was developed for, I believe it was basidioblastoma, where you have extremely high levels of insulin. So you develop hypoglycemia. As a consequence of hypoglycemia, you develop neuroglycopenia. So there was this drug called diazoxide that was developed that suppresses insulin secretion. And that then results in an increase in blood glucose. And it's early days now, but it seems to have worked quite nicely in a couple of patients. So basically what you do is you increase glucose, blood glucose to anywhere between 120 and 180 milligrams per deciliter. That increase in the blood glucose then forces more glucose into the brain. It addresses the neuroglycopenia that you find in this disease. And that seems to have worked in some small subset of patients.
Two questions on the floor. This we'll move on. We are actually running out of time. Good.
Thank you, sir. Very good talk. I have two questions. One is how this GLUT1 is involved in angiogenesis process. You showed some Filipportia and some extensions, but at molecular level, how this particular transporter is involved in angiogenesis process. That is one basic question. The other one is how you are going to take it further for translation in human, because you have done the preclinical work. What is your plan for translation?
I think both excellent questions. So, how does reduced GLUT1 result in this diminutive brain microvasculature? Some of the answers to that question come from work that has been carried out in the field of cancers, in particular work carried out by Peter Carmeliet. And what he has found is that endothelial cells are exquisitely sensitive to glycolytic levels, to glycolytic activity. Even a 30% knockdown of glycolytic activity in the endothelial cell is sufficient to cause these cells to stop dividing, to senesce, and eventually to degenerate. You can easily imagine that we are depleting GLUT1 much beyond that 30% of reduction that is required to really affect the functioning of these endothelial tip cells. So the bottom line is that it's because of the disruption of glycolysis in the endothelial tip cells that then results in this diminutive brain microvasculature. That's what seems to be happening in these cells. The second part of the question is, okay, so we've done this preclinical work. Where do we go now? And you know, it's been a bit frustrating for good reasons and for bad reasons. The work that I showed you depended upon a particular serotype of AAV. AAV9 worked quite nicely. Okay, and you know, in retrospect now, it all makes sense, but I think we were rather naive to think that, okay, the AAV9 works in mice and it's worked in the context of spinal muscular atrophy, then it's going to work in the case of GLUT1 deficiency syndrome as well. But what's become clear is people who work on these AAVs have found that they have quite different properties. And if AAV9 transduces the brain microvasculature in a particular species, it doesn't necessarily mean that it's going to do the same in another species. And we found out the hard way that AAV9 is actually not so good at transducing the primate brain microvasculature. Okay, so it doesn't matter what we've done in the mouse. We want to treat the human, so AAV9 is useless. So then what? So then we had to go through this long process to identify alternative serotypes. And people have been doing this. I mean, people like Viviana Grabnaru at Caltech and Ben Diverman at MIT, etc. And so they come up with these serotypes that they find transduce the murine brain microvasculature with amazing efficiency. And so they published these papers, and it says it's been used in the context of C57 black mice. So we adopted these. We brought them into the lab, used them on C57 black mice. Results look exactly like what's been published. When you change it to the FEB mouse, it doesn't work. You try it in the primate, doesn't work. So does one screen for these in the rodent? What we had to do was to go into the primate And I mean, you could say as luck might have it, but I think we deserve this because, it really was quite a process and very expensive. I mean, I did not realize that a cynomolgus monkey is basically what I paid for my very nice car that is sitting on my driveway in the US. So these are very expensive studies, but we did them and we found a couple of serotypes that transduce the brain microvasculature quite nicely. And so where do we go now? I think we are at the point where we need to do pharmacological and toxicological studies. Again, those are not cheap. And so where have we stumbled and what is stopping us from moving forward? In one word, it's money. To run a basic pharmacological and toxicological study under GLP, GMP conditions is going to cost us not less than $1 million. That's what we need.
I think the US time to close the session. I think this has been very inspiring. One of the things which I found, thank you, Sankita, for chairing the session. One of the things which I found so enticing about your talk is that you talked about your successes, but you also talked about your failures, and not every scientist does that. Hats off to you. Thank you so much. Let's all rise and clap my hand. Thank you.
I think a healthy dose of humility would do us all, whether it's scientists or whether it's politicians, in every walk of life. I think a degree of humility, I think, is always good.
So thank you so much, and we look forward to further interactions between you and the team from Neurology as well as the team from Stem Cell Center. Join us for a cup of tea and a little walk around the center. Thank you.
Thank you very much.